Good morning. Uh, in this session, all of you have to write down the notes on a role of induced polyploidy in plant improvement. A role of induced polyploidy in plant improvement. In polyploidy, application of allopolyploids, ALLO, POLYPLOIDs, has major role in crop improvement. There are three major applications. They are: 1. Transfer of characters from one species to another. 2. Production of new crop species. 3. Widening of the allopolyploid crop species. Next paragraph. The amphidiploids, AMPHIDIPLOIDs, amphidiploids are used as a bridge, bridge, to transfer characters from one species to a related species, that is, from wild species to a cultivated species. So, the use of amphidiploids had become necessary between cultivated and wild species. The amphidiploids are produced in increasing the number of F1 interspecific hybrids, and it is crossed to the recipient species, RECIPIENT, recipient species. The progeny of the cross become amphidiploids. It is used in backrows. Next para. The amphidiploids are used as bridging species, BRIDGING, bridging species, in tobacco varieties, TOBACCO, tobacco varieties, comma, Gossypium, GOSSYPIUM, Gossypium species, etc. Next subheading, creation of new crop species. Allopolyploidy, ALLO, POLYPLOIDY, allopolyploidy, enable to create new species. This species would be superior to the existing crop species. Some of the present day important crop species are allopolyploids. There are existing as well as new allopolyploids are synthesized in the same manner as they are produced in nature. So, an expected duplication of nature's NATURE nature's own OWN method would lead to the creation of new and superior crop species. First up, next para. Examples in which successful synthetic allopolyploids produced are subheading 1. The tetraploid or hexaploid wheat. Tetraploid or hexa, H-E-X-A, hexaploid wheat. Example, wheat into rye, W-H-E-A-T, wheat into R-Y-E, rye, gives triticale, T-R-I-T-I-C-A-L-E. First up. Next paragraph. This cross show the desirable features such as yielding ability and grain, G-R-A-I-N, grain quality. First up, the superior quality of triticale is produced by intensive research for about 50 years. First up, next subheading 2, alloploids, A-L-L-O-P-L-O-I-D-S. Example, Raphanobrassica, R-A-P-H-A-N-O-B-R-A-S-S-I-C-A. Yes, write down. The triploid Raphanobrassica is obtained by crossing between Brassica, B-R-A-S-S-I-C-A, Brassica, and Raphanus, R-A-P-H-A-N-U-S species, in brackets, Brassica and radish, B-R-A-S-S-I-C-A, Brassica and radish, R-A-D-D-I-S-H, close bracket, full stop. The hybridization and the selection at this polyploid have shown effective improvement. Full stop. Next sentence. Varalakshmi, V-A-R-A-L-A-K-S-H-M-I, is another hybrid variety of cotton, C-O-T-T-O-N. First up, it is raised by interspecific hybrid between Gossypium hirsutum, Gossypium hirsutum, and Gossypium barbadens, Gossypium barbadens, B-A-R-B-A-D-E-N-S-E. Then 3. Widening the genetic base of existing allopolyploids. Widening, W-I-D-E-N-I-N-G, widening the genetic base of existing allopolyploids. Yes, write down. The natural existing allopolyploids are introduced in a variable manner. First up, example, Brassica napus, B-R-A-S-S-I-C-A, N-A-P-U-S. First up, it shows only the synthetic new allopolyploids First up, a crossing of Brassica campestris, Brassica campestris, with Brassica oleracea, Oleracea, Brassica oleracea, with the chromosome number N is equal to 10AA, capital A, capital A, and N is equal to 9CC, capital C, capital C. First up, the amphidiploid Brassica napus, NAPUS, is with chromosome number N is equal to 19, 19, AACC, capital A, capital A, capital C, capital C. Full stop. So this form an autotetraploid. Full stop. In this case, the crossing is very difficult, and for spreading, the embryo culture is used. First up, next para. Though THOUGH, though there are progressive benefit in plant breeding by allopolyploids, there are certain defects also. First up, example, certain allopolyploids show low fertility and exhibit undesirable features. First up, next para. The synthetic allopolyploids have to improve through extensive breeding at the polyploid level. First up, next sentence. It involves considerable time, labor and other resources. First up, next paragraph. Only a small portion of allopolyploids are promising. First up, thus a costly costly trial and error has to be done before one is likely to come across across a promising allopolyploid combination. First up, it can be improved through breeding to yield a new crop species. Okay, in this topic, we started with role of induced polyploidy in plant improvement, but the same will ask either as how chromosome manipulation help in plant breeding, or the role of individual level chromosome manipulation in plant breeding, or importance of induction of polyploidy in plant breeding, or historical aspects and genetic basis of modern plant breeding. Such a level this topic may ask for your study or examination process. Okay, thank you.